and then we'll get into full-sib method and perhaps even into the S1 method. So we'll see how far we get. But by way of review, we began with the simplest half-sib method, which the corn breeders call ear-to-row selection, because in the first season you just simply allow plants to open pollinate. You harvest a seed from each individual plant, and that seed becomes a half-sib progeny. And so in corn, it would be like harvesting an ear from the plant, and the next season you're growing out the seed from that ear into a row. So it's called ear-to-row method for corn breeders. But this method can be used in all kinds of crop species, and I've actually used it in alfalfa before as well, just by harvesting the seed from individual plants there. So the second season you're growing out a row and basing your selection on the average of all the plants within a particular progeny, a half-sib progeny, to determine which are the superior genotypes or the superior parent plants that came from. Now, being an annual plant, the parent plant's not around any longer by the time you've done the evaluation in season 2. So what you need to do is take remnant seed, just some seed of each half-sib progeny that you've kept in seed storage somewhere in individual packages. You'll just take the remnant seed of those superior half-sib progenies and bulk those to become your cycle 1 seed. By the way, by way of review, what's the major advantage of a genotypic method over a phenotypic method? I actually gave you 2 of them, but one in particular I want you to keep in mind. One of them had to do with breeding value that we'll talk about in the quantitative genetics course next semester, but the other one is even more important to remember. What's the major advantage of it? And think about the disadvantage of a phenotypic method. Remember, with the phenotypic methods, with traits of low heritability, there might be so much environmental variation in the field that you're doing your selection in that it's difficult, if not impossible, to determine which are the superior genotypes. So with the genotypic methods, like the half-sib progeny method, it allows you to do what? To compensate for at least some of that. Yes? Can you speak up just a little bit louder? Exactly. So all the plants within a particular half-sib progeny, you're basing your evaluation on the average performance of all of those plants, however many plants are within a row, and number of replicates within a field, and perhaps even over locations and years that you're evaluating these half-sibs. And so you're taking plants that are related to each other and evaluating them in more than one micro and possibly even macro environment. By macro environment, I mean, different locations or different years. Okay? So there's less influence, at least you hope there to be less influence of the environment on you as a plant breeder's ability to identify the superior genotypes. Okay, so that's the major objective of these genotypic methods. Now, with this particular method, one of the disadvantages, I'm skipping a few slides because we already covered these. One of the disadvantages is that there's not as much genetic variability among the progenies, plus you do not have pollination control because pollination's already occurred before you do the evaluation in season 2 among the half-sib progenies. So how can we actually double this value? It's at 1/4 with the method we just talked about, but then we modified this to talk about the next method by doing what kind of modification so that we can get pollination control and be selecting the male as well as female gamete. It's the same thing we did with phenotypic selection. Anybody remember what it was? Yes? Okay, exactly. So you need to self-pollinate each plant as well. So there needs to be cross-pollination occurring so that you can, if you're evaluating yield, you need the grain to evaluate the yield on, but also on each plant you need to self-pollinate it. So that's what we did with this modified half-sib family selection. Again, in the first season, we just allowed for open pollination, but we also self-pollinated each individual plant. In season 2, we evaluated the replicated half-sib progenies, but instead of going back to bulk remnant half-sib seed, now we're going to take the S1s of the parent plants that produce the superior half-sib progenies. So again, we're evaluating half-sib progenies, but we're going to intermate the S1 progenies of those superior plants. So in season 3, then we grow out the S1s and allow those to interpollinate so that we can get what? What are we trying to do when we do that? We're not only producing the cycle 1 seed, but the objective of the interpollination among those selected plants is what? So that you can have what the next generation? Okay, you want to get genetic recombination there. Hopefully you'll get new recombinants from those plants so that the next generation you can identify even better plants than you did in the first generation. Okay, so it's for genetic recombination. Any questions on the scheme? We went through this a little rapidly at the end of the last period, and this is really where we stopped. Okay, so for advantages, compared to the original half-sib method that I covered first just a few minutes ago, you theoretically make twice as much progress on selection because you now have pollination control. You're selecting the female and the male gamete. Therefore, this A value is going to be what? Anybody remember what it was before? Yes? Exactly, 1/2. Before it was 1/4, if you happen to remember that. Because of pollination control, we're doubling it, so it's now 1/2. Okay? Disadvantages. Anybody think of any disadvantages? It's starting to become a recurring theme with some of these disadvantages. Yes?
More work.
Yeah, exactly. It's more work. It's a lot more work to accomplish, and thus more expensive to accomplish as well. Because now, you not only to allow open pollination to occur, which in a cross-pollinated species is easy, you just let nature take its course there, but you also have to self-pollinate each individual plant. You have to, in season 2, grow out the half-sibs and evaluate them. In season 3, you have to grow out the S1s and interpollinate them and harvest them and all of that. So it is a lot of work, and so you have to consider, is it worth the work to make the, to get the progress and selection that you need? And in some cases, it might be, the answer might be yes. In other cases, you might decide no. It does take 2 cycles or 2 years to accomplish a cycle of selection unless you can grow out 3 seasons in a single year. So if you do have a winter nursery, again, I'm using the example of New York State where we have just a single season, which is a relatively short season, growing season most years. But there is just a single season to grow out plants. So in a winter nursery, you would have to have 2 seasons. And I asked our corn breeder, Margaret Smith, a few years ago. She actually has a winter nursery down in Florida, and I asked her if you can actually get 2 seasons down in Florida to produce seed, and she said yes, you can if if you real quickly in the first season down there harvest the seed and make the turnaround real rapidly and replant it to go through the second season. So it's doable, but it's really tight. It's a tight time frame, but you can get 3 seasons in, 1 here and 2 down there. Given that, then, let me go back to the principle we talked about before. Which of the 2 seasons up here would you put in the winter nursery? Another way of asking is which season would you do up here? Yes?
1 and 3 in the winter.
Which one again?
1 and 3.
Would be in the winter nursery, okay? Seasons 1 and 3, because these are the 2 seasons that all you are doing is just producing seed. You're growing up the plants and allowing them to interpollinate and producing seed. Remember season 2, whichever season, I don't want you to memorize that season 2 is the one you do in New York State. What I want you to remember is that whichever season that you are doing your evaluation for the trait, that's where you do at your home base. And in this case, it is season 2 because that's when you evaluate the half-sib progenies. Okay, everybody sees that then? And that's clear? Okay, great. Okay, and the third one, again, could be an advantage or disadvantage, just like we talked about with simple recurrence selection for the phenotypic method a few times ago, that since you need 1 ear in corn for the cross-pollination and 1 ear for self-pollination, you do need prolific plants, more than 1 ear per plant. So that could be a disadvantage, or it could be an advantage, again, because prolificacy is related to yield in the first place, so it could actually help you to boost yield by being forced to use those plants in the first place. So it just depends how you look at it. Okay, any questions on that last method at all then? By the way, it dawned on me last night when I was thinking about this that I might be rushing through some of these slides for advantages and disadvantages, and I sort of forgot that you have to write that down because it's not in your notes. So if I flip too fast, just kind of yell out or raise your hand so I can slow down. Or I guess these are up on the web anyway, and you can take a look at them on your own as well. But I'd be glad to slow down a little bit if you're trying to write. Okay, here's a third type of mass or of half-sib selection, another modification. And this one's substantially different than the other two that we've done. You start out in season 1 by just having open pollination to produce your half-sib seed, but in contrast to the last method, you do not need to self-pollinate any of the plants. This one has no self-pollination involved, just cross-pollination there. Okay, so that's an advantage in a sense. In season 2, you're going to do two things. Now, remember a time or two ago, we said, in fact, it was the last period on Monday, and I had you stand in two different parts of the room. You represented two half-sib progeny rows. And I mentioned to you that although there's substantial variability among the half-sibs, that there's a lot of genetic variability among plants within the half-sib rows. So if you add the two, the additive genetic variation among half-sibs plus the additive genetic variation among plants within a half-sib row equals the total additive genetic variation among the individual parent plants in a population. Well, when you're doing half-sib selection, one disadvantage is that there's only theoretically 1/4 as much additive genetic variation among the half-sib progeny rows. The rest of it's within. So when you do half-sib family selection, you're only capitalizing on 1/4 of the additive genetic variation in a population. So that's a major disadvantage. With this modification, Lonquist, back a few decades ago, developed this method which capitalizes on both types of the additive genetic variation in both types there, both among and within. So it's really a combination of two breeding methods into one. It's a combination of half-sib progeny testing and also mass selection or phenotypic selection. So it's both combined into one. So again, you're capitalizing on all the additive genetic variation in a sense. So again, in the first season, you just allow open pollination to occur to produce the half-sib seed. In season 2, you put out two types of trials in a sense. One is an evaluation trial, just a replicated half-sib evaluation, just as we've discussed before. Okay, so that's the place where you're evaluating the average performance of the half-sib progenies and identifying the superior half-sibs. At the same time, in season 2, separate from these evaluation trials, in fact in an isolated spot, you have what's called a crossing block. Now in that crossing block, there's two things that you're trying to accomplish. One is to get a good random cross-pollination among the half-sib progenies to produce the next generation of seed, and I'll tell you more specifics about how that's done. But the second thing that's done there is the mass selection. That's where you're identifying the individual superior plants in those half-sib progenies. So, again, to put the combination of the two together, you would evaluate the half-sib progenies in this replicated evaluation trial, and then once you've identified the superior half-sib progenies, then you go into the crossing block, and only in those superior half-sib progenies, again as identified over here, then you come into here where you've got one replicate of the same half-sib progenies. You go within those superior half-sibs and identify the individual plants that are superior. So you're doing the mass selection within the superior half-sib progenies. Does everybody follow that? It's a real mouthful to try to get that out. Everybody's following that pretty well? Okay. Now, the other thing that you're trying to do with the crossing block, as I said, is to get a good random cross-pollination, because when you plant out this crossing block, and again you have to plant it out as half-sib progeny rows, because you're only going to do mass selection within the superior half-sib. So you have to keep the half-sib separate. Okay, but when you have rows like that, if you have like 200 or 300 of these rows across the field, you're not going to get very good random pollination, because rows number 1 and 2 will pollinate real well with each other, but they won't pollinate much with rows 200 or 300 way down at the other end of the field. So to compensate for that, what Lonquist recommended is that every 4 rows, I don't know if you can see it up here or on your handout, but there are 4 rows that are a little bit lighter gray and then 2 darker rows next to it, and it keeps alternating like that. The 2 darker rows are a mixture of the same half-sib progenies. It's the same population, but you take a little bit of seed out of each one of the 200 or 300 or 400 half-sib progeny rows, or out of the bags or envelopes or whatever they came from, and just mix them together so you have a bulk of the population. That's what those 2 really dark, bold colored rows are there. So in corn, these 4 rows that are the half-sib progeny rows, you detassel them so that they do not contribute pollen. Only allow pollen to come from the darker rows where it's the mixture of the population. And thus you get a better random pollination on each one of those half-sib progeny rows. Okay, does everybody see how that works then? Yes, Andres?
So in the crossing block, you only have those half-sibs that were selected to be the best.
Well, in this particular scheme, that actually is a modification of what I'm talking about, so let me get to that in just a moment. But in the original scheme, this crossing block is grown out at exactly the same time as the evaluation trial. So you have the superior ones there, but you have all the other ones there too. And that becomes a disadvantage in the method because when you allow cross-pollination, you're getting cross-pollination from the undesirable as well as desirable half-sib progenies there. So it is planted out at the same time, but you only do your mass selection or phenotypic selection within the superior half-sib progenies. Yes?
I don't understand if in the crossing block you're selecting for the superior individual plants, why do you have the half-sib evaluation trials going on?
Because in the evaluation trial, this is a replicated trial that is to give you a better evaluation of the performance of the half-sib progenies. I mean, that's a good question, and in fact, I've had that question in previous years. Why can't you just do it all in one single trial, in this one or that one, maybe this one, since this is better cross-pollination? And the answer is that you could do that if the trait that you're evaluating is one that you're going to evaluate individual plants anyway. If it's disease resistance, as an example, you might be evaluating individual plants and giving them a score or some kind of a rating anyway. And so you could do them both in the same time, in the same trial there. But if you're using this method for yield, you most likely would not want to evaluate yield of individual plants because it would take you forever to get it done. So what a breeder would do is go down through with a small plot combine and just cut the whole row to get the total yield of the row. Okay, so if that's what you're doing, you do need to have 2 separate trials because as you go down through the combine, you're not evaluating individual plants, you're evaluating the whole row in a sense. So in that scheme, you would have 2 different trials, one to do your yield evaluation, and then come in here, and only of the superior half-sibs that you identified over here, you come into the same ones over here and do your mass selection, where you do just evaluate individual plants. But, you know, both of you raised really good points there, and there are modifications that in some cases you might be able to accomplish in a more efficient way than what I'm describing as the classical method here. Now, Andres, you mentioned about, you know, does this crossing block do you only grow out the superior half-sib progenies? A few years later, a couple of authors by the name of Compton and Comstock, I don't know if any of you have ever heard Comstock's name. I'm not even sure he's still alive. He was pretty old when I took a course from him at the University of Minnesota. But back in those days, he was a pretty famous quantitative geneticist. Compton was one of his graduate students, and they published a paper in Crop Science, I think in 1976, something like that. And what they recommended as a modification to this, because of the lack of pollination control, is to delay growing out the crossing block until another season and only grow out the superior half-sib progenies. And so what that's going to do is to double part of the formula. In fact, let me flip to this. Well, let me do it later. It's about 3 slides down the road here before I can explain it fully. But when you do that method, in essence, what you're doing is you're not totally doubling your progress from selection, and I'll have to wait until I get to that slide to show you why. But it's taking you twice as long to accomplish because now you have 2 seasons that you have to do at your home base. Both the evaluation trial and the crossing block you do in New York State if this is where you're doing the breeding. And so it's going to take you 2 years to accomplish in contrast to 1 year as this scheme is presented up here. And so I've never really been able to see the advantage when you think of the progress per year. But let me show you that when we get to the response from selection formula, and I think it will be clearer. So you all are bringing up some really good points and questions. Any other questions about the scheme or the methodology before we go on? Okay, good questions and good comments there. Here's what Wiebel and Lonquist. Wiebel, I think, was a graduate student of Lonquist, so they actually tried the method as you would expect them to do. They started out with 190 progenies of corn, and they selected the top 20% of the half-sib progenies, which works out to be 38 of them. So then within a crossing block, out of those 38 half-sib progenies, they selected 5 superior-looking plants within those half-sib progenies. Okay? And then, of course, as a crossing block, open pollination is occurring there, so they just simply harvest the seed off of that individual plant that they've identified within that superior half-sib progenies, and that becomes the half-sib progeny for the next cycle of selection. So you've got 38 progeny rows times 5 plants within a progeny row. That works out to be exactly 190 progenies the next cycle of selection. So you have the same number of progeny rows from cycle to cycle when you do it that way. Everybody see how that worked? Okay. You might, if you're interested, you might actually get into crop science and read this paper. It's kind of an old one now, but it's interesting about the way they logistically actually accomplished it, because they were actually doing the mass selection, as I recur, by visual estimate, but they didn't actually identify the superior half-sib progenies quite yet, so they actually took out some red paint, spray paint, and painted the tip of the ears that they wanted to actually save once all the data were collected. So it's kind of interesting the way they did the logistics. As a result, they went through 4 cycles of selection, and they were able to realize a 10% gain in a selection in terms of yield for each year, which amounts to a cycle of selection. And this is compared to about 4 to 6% increase in yield from simple recurrent selection. Remember, that's the phenotypic selection where they produce the S1s and interpollinate the S1s there. Keep in mind, as you read the literature, and breeders are notorious for coming up with a new modification of a breeding method, which is fantastic, but beware of reading in the literature where they're comparing the success of one method with another, because in this case, their 4 to 6% from simple recurrent selection was from the literature. You know, they were just averaging what other people have done. But it's really comparing apples and oranges, because if you're really going to do a good comparison of breeding methods, you need to be doing at least two different breeding methods in the same plant population for the same trait, in the same locations, field, environment and everything, to have a valid comparison among breeding methods. So this is just kind of a little bit, I don't blame them for making the statement, but you have to be careful to take it with a grain of salt, since it wasn't actually comparing apples and apples in a sense. Okay, now this is where I want to get to with this, one of the advantages, the main advantage, is that you're combining 2 breeding methods, half-sib and mass selection, and capitalizing on the additive genetic variation in both types of plants there. As a result, it gives you a complex response from selection equation. And so you've got a part here in this first row, this first line, which is your response from selection for the half-sib, and thus I have the phenotypic standard deviation for half-sibs, which is what HS stands for. The next line there is for, I have sigma sub W. That means the standard deviation within a half-sib row. In other words, among plants within. That's the phenotypic or the mass selection part there. Okay. Notice 2 things. One is that my selection index, I have an I and an I sub 2. You're probably going to have a different selection intensity for among versus within. Okay, so that's why I've got a different I there. And the other that's most noticeable is your A value. It's 1/8 for the half-sib and 3/8 for the mass selection. Now, part of that, the reason that it's low is because of the lack of pollination control, but the other part is becoming less intuitive. It's getting too complex for it to make sense at this point in time. So you just have to kind of take it by faith that that's what the A values really are. And if you're like me, I'm pretty distressed by seeing that that A value is so low there. But if you do like Andres was talking about, and that is you wait another season and just grow out the superior half-sib progenies before you grow out the, before you grow out the crossing block and do the mass selection, then your pollination is only among the superior half-sib progenies. So you can double this part to 1/4, which is better, but again, it takes 2 years to accomplish. So if you double this part but not double that part, you're not doubling the total response from selection equation. And therefore, it's taking you twice as long, but you're not getting twice as much value out of it. And therefore, your response per year is actually less if you do that than if you do the original method where you've got the crossing block grown at the same time as the half-sib. Does everybody follow that? Okay. So as many times as I've read Compton and Comstock's paper of their modification, I hadn't really figured out why it's really an advantage, at least when you look at the theoretical response curve there and think about the time involved as well. Okay. Yes?
Can't you grow the crossing block in the winter greenhouse or the winter area?
Yep. You could do. Yep, you could do that if all you're interested in is just cross-pollination, but since you're doing mass selection, and if it's trait-like yield, you don't want to do that in a greenhouse or in a winter nursery because what you select as being superior in yield there in Florida or Puerto Rico or Hawaii is not going to be the same as yield up here in New York State. But if you were selecting for some trait that would be the same, and I don't know what it would be, maybe a disease or insect resistance, then you could do that. Okay, but good. I'm glad that you all are thinking. You're thinking with good reasoning there, in all of this. Okay. Some disadvantages. It's a little bit more complex breeding method than some of the others, but that doesn't slow us down most of the time. You've got both the half-sib and the mass selection to deal with there. And again, pollination is from unselected as well as selected plants. Even if you were to delay your crossing block until the next year, and therefore you're only growing the superior half-sib progenies, okay, you are getting only the selected pollen from the selected half-sibs, but still there's pollination among the undesirable plants within those, and so you're not getting only pollination from the mass-selected plants, the phenotypic selected ones. So even the second part of the equation is still not going to be doubled because you still have a lack of pollination control even in the crossing block there. Okay, any questions before we go on to another method? That's probably one of the most complicated ones that we'll hit before we get to reciprocal recurrent selection on Friday. Okay, this is the last half-sib method that we're going to cover. And this one, with half-sib progeny selection using a tester, you can use it for two different purposes. If your objective is to produce really good inbreds for hybrid variety production, and you want to identify the superior plants that will nick well together for a hybrid, you want your tester population, or the population you're using to cross with the one you're trying to improve, you want your tester to be a real good one, because your tester population, you'll be extracting good material out of that, as well as the other population to make a hybrid variety. So that's one objective. But we'll actually be talking about that in more detail when we talk about reciprocal recurrent selection on Friday. My objective in using this method right now is what we talked about the first day that I think it was the first or second day I was with you last week, where we had the situation where you have a lot of dominance in the population for that trait, and you cannot distinguish phenotypically between homozygous dominant and the heterozygote. If you have a situation like that, and it's really important for you to be able to distinguish those, then one method of doing that is with this half-sib family selection using a tester. So in this case, what kind of a tester do you want? The tester you want to use, as we spoke last week, is going to be a recessive one, okay? But does that mean it's going to be a good performing population or a poor performing population that you use as a tester? Yes?
Would it be poor because you want the performance of the progeny to be totally attributed to the plants that are not the tester?
Good way to say it. You want a poor performing tester. In fact, the poorest performing tester you can get that will work, the better. because as you said, you want to be able to evaluate what the good genes are in the population that you're trying, what we call the target population, the population you're trying to improve. Okay, so when I actually tried this method before as well, what I did is try to get a hold of the rattiest-looking alfalfa population I possibly could, the lowest yielding one that I could possibly get a hold of to use as a tester population. So here's how the method works. The schematic is a little confusing. These dots up here that have the circled X's by them are the plants from the target population, the population that you want to improve. These rows of 4 that are vertical are the tester population. Those are the poor performing population that you hope is in the recessive condition. Okay? So the way that you accomplish this is take a collection or mixture of pollen from the tester population, probably from more than 4 plants. I mean, for simplicity on the diagram, I'll only put 4 there. But just take a whole bunch of plants and collect pollen from them, mix it up, and then go in and pollinate each one of the plants of the target population up here. Okay, so what that does when you harvest the seed of that test cross, that is a half-sib progeny that you've already harvested there. Okay, the seed are half-sibs of each other, because again, it's a collection of pollen from the tester population. So in season 2, you grow out those half-sib progenies of the test cross and identify the superior ones, but you do not take remnant seed of those half-sib progenies and bulk that for cycle 1. Why don't you do that? What would happen if you did that? Okay. I think it's pretty obvious to most of you. Yeah, you'd essentially make one step forward and about 2 or 3 giant steps backwards, because remember, half of the genes in those half-sib progenies are coming from the tester population, the tester, okay, and that's in a recessive condition, so you're carrying those genes down into the half-sib progenies there. So you're really taking a lot of steps backwards, even though you're trying to attempt to step forward with it. So you go to our standard method there of producing self-seed on each one of these plants in the target population. And then in season 3, you're growing out the selves of those plants that produce the superior half-sib progenies. And in season 3, those selves get interpollinated to get genetic recombination and to produce your cycle 1 seed. And then you can start the process all over again. Questions on the methodology there? Does everybody follow it? Okay, everybody's pretty clear? Okay, anybody think of, well, let me go first of all to the advantages, but while we're going through the advantages, think of any disadvantages that come to your mind really quickly. Looking at it genetically like we did last week, you know, again, if your target population represented by these 3 possible genotypes, the homozygous dominant, heterozygote, and the recessive there. The recessive should be pretty obvious, but if you're using recessive tester population, then this will be genetically what you'll have. All of the progenies of this homozygous dominant will be really good if there's complete dominance for the expression of the trait. Okay, so all the progenies will be good. The heterozygote will be a mixture, a 50-50 mixture of good and poor performing. But if you take the average performance of that, it will not be as good as the test cross with the homozygous dominant there. And of course, the recessive are all going to give you recessive progenies and therefore be poor performing. So it helps you to distinguish between the homozygous dominant and the heterozygote when you have a lot of dominance for the expression of the trait. Now, in reality, what a lot of people will do is that if your trait is heritable enough, you can make plenty enough progress from selection with phenotypic selection, and then like that one response curve I showed you last week with bacterial wilt on alfalfa where it plateaued off, that was where it started to plateau. We probably had the population performance good enough in alfalfa where we didn't need to go farther, but if it was important to get off of that plateau and make even more progress, then I might have to resort to this selection method to be able to distinguish between homozygous dominant and the heterozygote to make any more advanced, or to use molecular techniques like molecular markers to be able to do that. Questions on this then, on the advantage of the method? Okay, what can you think of in terms of disadvantages then? One that's kind of a recurring theme, as I mentioned before. Yes?
If you don't have complete dominance, then
Okay? Yeah, if you don't have complete dominance or even a lot of partial dominance, it probably won't work. It won't be an advantage. But it will also be a lot of what else? It's a lot of work, which is what my question mark is there. Plus, it takes 3 seasons to accomplish. Okay, but again, you might be able to get it done in 2 years if you can get two seasons worth of seed production done at a winter nursery there. Okay, but it is a lot of work because both types of progenies you have to do by hand. You have to make the test cross by hand. So the half-sib progenies, in contrast to the other half-sib progeny methods, where in a cross-pollinated species you can just let open pollination to occur by nature. Now, because it's a test cross, you have to do that cross by hand, plus you have to do the selfing by hand. That gets to be a lot of work to do that, plus all the work it does to grow out both types of progenies in the 2 different seasons there. So it is a lot of work to accomplish, probably more work than any of the other methods we've talked about so far. But again, if it's important for you to distinguish between the homozygous dominant and the heterozygote, then it's a method to try to use there. Okay, another method. We're going through a lot of them today. This one is a lot simpler, the full-sib progeny test. With the full-sib progeny test, you're not allowing open pollination to occur because that would produce half-sib progenies. Now you have to, by hand, take 2 plants at a time and cross-pollinate them both. You can do reciprocals as well between the 2 plants. So by producing seed on 1 plant crossed with and only one other plant, you're producing a full-sib progeny where all the offspring are going to be related by having both parent plants in common. Okay, so you do a bunch of those full-sib crosses from your population. In season 2, you grow out those full-sib progenies in a replicated trial and evaluate them and identify the superior ones. And then you take the remnant seed. You keep some of the full-sib seed in packages and seed storage. Take the remnant seed of the superior full-sibs and just bulk those to be your cycle 1 seed. Now, when you do it that way, you haven't had a lot of genetic recombination occurring because there hasn't been a random pollination across the population. So you could take your remnant full-sib seed and plant that out and go another season and allow cross-pollination to occur to get more chances of recombinance. But I don't think that most people who use this method go that extra step. They just figure that the cross of 1 plant with 1 other plant is good enough and it's not worth the extra time and the work to get better recombination or more random pollination by growing out another season there. But you could do it either way. Okay, questions on the methodology then? It's really conceptually very simple compared to the others that we've talked about recently. Okay, in terms of advantages, theoretically there's twice as much additive genetic variation among the full-sib progenies as there was compared to the half-sib progenies. So with the half-sibs, I said there's 1/4 as much additive genetic variation compared to the parent plants that came from. Well, in the half-sib, there's half as much compared to the parent plants, or compared to the half-sibs, there's twice as much compared to that. So the A value is 1/2 instead of 1/4. Okay, so that's a major advantage for the full-sib progeny test. So if you have a heritability that's really, really low and half-sib progeny is not working, then you might try full-sib where you can capitalize on a higher percentage of the additive genetic variation. The more additive genetic variation that there is among the progeny rows, the easier it's going to be able to distinguish among the progenies there. There's just more variability there, and it will be easier to identify the superior ones. Another advantage, which is kind of minor but still an advantage, is that for every half-sib progeny row, you're really evaluating 1 parent plant's performance, or genetic potential rather. With a full-sib progeny, you're evaluating twice as many parent plants, because for each progeny row, you're evaluating the average genetic potential of 2 parents. So you can involve twice as many parent plants in essence. Okay, disadvantages. When you do a full-sib progeny test, what would happen if you had a really, really good, genetically a really good plant, parent plant, that happened to be crossed with one that was not very good at all? The full-sib progeny would probably be mediocre. You might not select it. So there is a chance that when that happens, there is a chance that you could be discarding some really good plants genetically. Okay? So for that reason, what some people might do is actually do a little bit of phenotypic selection to select out your parent plants that you're going to do your full-sib crosses with. But even at that, there can be some occasions where you could be discarding some superior plants just because it by chance was crossed to a really poor plant to give you a mediocre half-sib progeny. And compared to some of the half-sib progeny tests, it is a little bit more work in the sense that you have to do all the crossing by hand, but maybe that's only in comparison to the first half-sib progeny test we talked about, because some of the others are more complex and a lot more work anyway. Okay, any questions on that method? Man, we're really cruising through this. I'll probably have you thoroughly confused because we're covering so many methods today, but it means on Friday we'll be able to cover fewer of them. Okay, let me cover, we've got time for one more and then we'll quit. And this has probably got you more than saturated with methods here. The S1 progeny test. Now for this one, you do not make any crosses. There's no open pollination occurring here, at the beginning at least. You have to go into each individual plant in your population and self-pollinate it. So you need to do that by hand. The next season, you're growing out replicated S1 progeny rows and doing your evaluation for the trait, like yield. Again, just like all the other progeny rows, looking at the average performance of those of each of the half-sib progenies there, or S1 progenies rather. Okay, so then once you've identified the superior S1 progenies, you go back in your seed storage and take remnant seed of the S1s. So again, you have to keep some remnant seed of the S1s and not plant them all out in season 2. You take the remnant seed of the superior S1 progenies, grow those out in season 3, and so in season 3 you're just allowing for open pollination to occur to get your genetic recombination. Okay, everybody follow me on the concept then? Okay, so no cross-pollination occurring at all until you get to season 3 there, and you're evaluating S1 progenies. Major advantage in this is that there's actually more genetic variability among the S1 progeny rows than there was compared to either the half-sib progeny or the full-sib progeny row. Okay? In fact, theoretically, there's just as much additive genetic variation among the S1 progeny rows as there was among the individual parent plants that produced them. So you didn't lose any of the additive genetic variation there. So your A value in the response from selection curve is going to be 1, which is exactly the same as it was for most of those phenotypic methods that we looked at. But now you have the advantage of being able to look at related plants replicated over micro and macro environments. Now there's another factor in here too, which is a capital B, but in reality it's supposed to be written as a Greek beta, but I didn't have a Greek symbol on my computer when I did this, so that's actually a beta. And what that beta is, it only exists if there's any dominance for the trait in that population, but it is not dominant genetic variation. It's if you were to look at the actual equation for beta, it would be some long thing that didn't make a lot of sense, and in reality, in most cases, you can't even compute what the number is. But it is there, meaning that if there is some dominance, then it might give you a little bit of kick in yield or whatever the trait is, to the extent that this beta might be important. Again, it only exists if there's dominance. If there's no dominance for the expression of the trait, beta 0. But I don't want you to worry about beta too much there, just to be aware that it's there. But the important thing is that this A value is 1 for this S1 progeny method. Another method is that it allows you to uncover deleterious recessive alleles. Sometimes you can see some deleterious stuff that comes out when you cross-pollinate, but most of the time it gets masked by a dominant gene. When you self-pollinate it, you get more recessives that segregate out, and therefore you can identify some of these deleterious recessives when you look at the progenies. So it's easier to see those, whereas they're masked with cross-pollinations there. Actually, I've done this with alfalfa before, where when you look at S1 progenies, you really see some weird-looking stuff when you grow seedlings in a greenhouse. You see everything from from albino seedlings. A lot of chlorotic ones come out. You see different leaf morphologies that you don't see when you cross-pollinate. There's all kinds of weird stuff that you see when you self-pollinate, especially a crop like alfalfa. So it's really kind of interesting when you do that. I got to thinking last week that we're going to start selecting for a disease resistance called spring blackstem in alfalfa, because there was an awful lot of it around the state this past season. And we did a little bit of research with it. Back about 15 years ago, I had a graduate student working on a master's program, a student from Spain, and she actually compared two different breeding methods for spring blackstem and found out that the heritability was too low for phenotypic selection. In fact, she even tried 2 cycles of phenotypic selection and made absolutely no progress on selection. But she also did a study where the heritability with the Half-sib method was significant. So it told us that if we're going to make progress on selection, we're going to have to use a progeny method. So I got to thinking the other day, as I was thinking about this class, that actually if we did an S1 progeny method, it would probably be even more successful than a Half-sib, because I've got a whole lot bigger A value than I would for Half-sib method. So we're actually going to try that over this next year and use the S1 progeny method to see if we can make more significant progress on selection for this one disease resistance that tends to have a pretty low heritability. Okay, and let me finish up by talking about disadvantages, and then we'll quit unless you have some questions. This method is a little bit more work to accomplish, again, especially compared to the original Half-sib method, because with this one all the progenies have to be made by hand. Okay, compared to some Half-sibs, it's maybe simpler amount of work, less work, but compared to the original one, it's more work because you have to do all the progenies by hand. And with this method, you're really not evaluating combining ability because you're not crossing it with any other plant. It's all self-pollination occurring. So if that's important to you, this method doesn't do it. Okay, we've covered a lot during this period. Let me ask if you have any questions before we quit.